What is mitosis? Well, it's simple. This is a cell, it divides into two. Ta-da! Mitosis has taken place. These cells that are formed are known as the daughter cells. But is it really that simple? Well, it's not. In this video, we are going to discuss the subphases of mitosis. But before that, you need to understand two terms: karyokinesis and cytokinesis. If you break down the word karyokinesis, karyo in Greek means nucleus, and kinesis means division. So, this is when the nucleus of the cell divides into two daughter nuclei. If we do the same with cytokinesis, cyto means cell, and again, kinesis means division. So this is when the cell itself divides. If you notice, karyokinesis makes sure that the genetic material is equally divided, while cytokinesis ensures the physical separation of the two daughter cells. Now that we're familiar with these terms, let's move on to see how they apply in mitosis. First, it's important to understand where mitosis actually fits within the larger context of the cell cycle. The cell cycle consists of two main phases: interphase and mitosis. Interphase is the preparation phase where the cell grows, duplicates its DNA, and gets ready for cell division. It has three subphases: G1, S, and G2. In the G1 phase, the cell grows and produces proteins necessary for DNA replication. During the S phase, DNA replication occurs and organelles like centrosome are duplicated. This ensures the cell has two copies of everything it needs for division. In the G2 phase, the cell continues to grow and checks are made to ensure the DNA and organelles are properly copied. By the time mitosis kicks off, the cell's got its organelles and DNA all copied and is all set to split into two. The subphases of mitosis are prophase, metaphase, anaphase, and telophase, which is immediately followed by cytokinesis. You can remember it by the mnemonic, please make another two cells. Now we're going to focus on two main structures, the chromosomes and the centrosome. Let's start with the prophase. In this phase, the chromosomes, which were previously spread out loosely, start to condense and become more compact. This condensation is really important because in later phases, when the chromosomes are pulled apart, their condensed form makes the process more efficient and manageable. Another thing that happens is the nuclear membrane, also known as the nuclear envelope, starts breaking down, allowing the chromosomes to be released. Meanwhile, the two centrosomes move to the opposite poles of the cell and the mitotic spindle begins to form. Now, what the heck is this mitotic spindle? Let's pause here to understand what this means. When we say that the mitotic spindle begins to form, it means that structures called microtubules start to extend from the centrosomes at opposite poles of the cell. These microtubules create a framework or spindle that will later help separate the chromosomes during mitosis. The spindle fibers attach to the chromosomes at specific points called kinetochores. The kinetochores are located at the centromeres. The spindle fibers attach here, making sure that the chromosomes are properly aligned to be pulled apart. The spindles gotta form to ensure the genetic material gets split right. Let's continue with the prophase now. So, to summarize, the chromosomes condense, the nuclear envelope disintegrates, and the spindle fibers begin to form from the centrosomes. Moving on to the next one, the metaphase. At this stage, the chromosomes line up in the middle of the cell, forming a straight line along an imaginary plane called the metaphase plate. This plate is located at the cell's equator, equidistant from the two poles. The spinofibers attach to the kinetochores of the chromosomes, and in the next step, which is anaphase, these spinofibers begin to shorten, pulling the sister chromatids apart. Each chromatid is dragged toward opposite poles of the cell. This step makes sure that the future daughter cells will receive an identical set of chromosomes, maintaining genetic consistency. Then we've got the telophase, where two nuclear envelopes form around the separated chromosomes. These chromosomes begin to decondense, returning to their relaxed, thread-like state. This phase brings us to the completion of karyokinesis, the process of dividing the nucleus. If you notice, karyokinesis isn't just something that happens in telophase. It's a step-by-step process that takes place across all the subphases of mitosis. Starting in prophase, the chromosomes condense. In metaphase, the chromosomes are aligned at the metaphase plate, ready for separation. During anaphase, the chromatids are pulled apart toward opposite poles. And finally, in telophase, the separated genetic material is safely enclosed into two nuclear envelopes. Once karyokinesis is complete, cytokinesis begins. The cell membrane starts pinching in, making a little groove called a cleavage furrow. It keeps tightening up until the cell splits into two daughter cells, each with its own nucleus and organelles. So this is how our cells divide. Through mitosis, they carefully split their genetic material to make identical cells. Each step ensures properly that the new cells are ready to function, just like the original. That's it for our video. Thanks for watching and have a good day.